What is hemoglobin? Introduction. Hemoglobin is an iron-containing protein in red blood cells responsible for transporting oxygen from the lungs to body tissues and carrying carbon dioxide back to the lungs for exhalation. Types of hemoglobin. 1. Hemoglobin A1. About 95 to 98% of adult hemoglobin is hemoglobin A1. It is composed of 2 alpha and 2 beta chains. 2. Hemoglobin A2. About 2 to 3.5% of adult hemoglobin is hemoglobin A2, which is composed of 2 alpha and 2 delta chains. 3. Hemoglobin F. Predominant type of hemoglobin found in fetus and makes up less than 1% of adult hemoglobin. It is composed of 2 alpha and 2 gamma chains. Normal developmental changes. 1. Fetal stage. Hemoglobin F is dominant hemoglobin in fetus, and HbA1 and HbA2 is almost absent. The reason is because HbF has a higher affinity for oxygen, allowing the fetus to extract oxygen efficiently from maternal blood containing HbA. 2. After birth. After birth to around 6 months of age, the gamma chains in HbF are gradually replaced by beta chains, and HbA1 becomes the dominant form. Genetic basis of normal hemoglobin. Alpha globin genes. Located on chromosome 16, these include HbA1 and HbA2, which code for the alpha globin chains. These genes remain active throughout life. Beta globin gene cluster. Located on chromosome 11, includes the HBB gene, which codes for the beta globin chain. These genes' expression is not active during fetal life and are expressed after birth. Gamma globin genes. These genes include HBG1 and HBG2 and are located on chromosome 11 and code for gamma globin chains. These genes are highly active during fetal life, but their expression is suppressed after birth. Delta globin genes. The delta globin chains of hemoglobin, found in HbA2, are encoded by the HBD gene, which is located on chromosome 11 within the beta globin gene cluster. Hereditary persistence of fetal hemoglobin. Genetic condition where HbF continues to be produced into adulthood. The effect is an increase in HbF that can be reached up to 100%. It is usually asymptomatic, may benefit patients with sickle cell or beta thalassemia. Hemoglobinopathies. Hemoglobinopathies are inherited disorders caused by mutations in the genes encoding globin chains of hemoglobin. These mutations can affect the structure or production of the globin chains. 1. Qualitative defects or structural defects, caused by point mutations that alter the amino acid sequence of globin chains. Sickle cell disease. Due to mutation in beta globin genes, there is substitution of glutamic acid by valine at position 6 of beta globin chain. Due to this mutation, abnormal hemoglobin HbS is produced. This abnormal hemoglobin polymerizes when there is low oxygen, and RBC's shape gets distorted, making it difficult for them to pass through small capillaries. Hemolysis occurs, causing anemia. Hemoglobin C, caused by substitution of glutamic acid by lysine at position 6 of beta globin gene. It is less severe than sickle cell, can cause mild hemolytic anemia. 2. Quantitative defects. These are caused by mutations that reduce or eliminate the production of globin chains. Diseases include all types of thalassemias. Thalassemias. Thalassemias are inherited blood disorders caused by mutations that reduce or eliminate production of alpha or beta globin chains. The severity depends on the number and type of mutated genes. Alpha thalassemia, when alpha globin chain production is reduced due to deletion or mutation in the HbA genes on chromosome 16. As a result, imbalance in the alpha and beta globin chains occurs, causing damage to the RBCs. Beta thalassemia. Point mutation or deletion of HBB gene on chromosome 11 causing reduced quantity of beta globin chain production. This results in excess of alpha chains and damages RBCs. Minor and major forms of thalassemias. The severity of thalassemias ranges from minor to major forms. Genetic bases of minor and major thalassemia. 1. Beta thalassemia. Beta thalassemia minor. Trait. Genotype. Heterozygous. 1 mutated HBB allele. Result. Slightly reduced beta globin production. Clinical impact. Mild or no anemia. Usually asymptomatic. Beta thalassemia major or Cooley's anemia. Genotype. Homozygous. Both HBB alleles mutated. Result. severely reduced or absent beta globin. Severe anemia requires regular blood transfusions. 2. Alpha thalassemia. There are 4 alpha globin genes, 2 from each parent. Severity depends on how many are deleted or mutated. Alpha thalassemia trait minor, genotype, 2 gene deletions. Result, reduced alpha globin production. Clinical impact, mild microcytic anemia, often asymptomatic. Alpha thalassemia major or HB Bart's hydrops fetalis. Genotype, all 4 genes deleted. Result, no alpha globin produced. Clinical impact, fatal death in uterus or shortly after birth.